In this video, I will be talking about the structure of DNA. So if you've ever seen maybe a model of DNA, you've probably seen it where it looks kind of like this. Kind of like two swiveled up lines. So we call this a double helix. So DNA is made up of a double-stranded helix. Double-stranded helix. So in this double-stranded helix, we have two strands. These strands are made up of backbones. What makes up these backbones is a phosphate and a sugar, a 5-carbon sugar. That's why it's in a pentagon shape. So there's a phosphate connected to this sugar, and they all connect in a line to make up the backbone of one strand. So let me label all of these. All right, so the same thing is on this other side, except it may look a little different because it is the opposite direction. So the other strand of DNA runs in the opposite direction. And I'll label these real quick for you. So something has to connect these two strands, right? What connects these two strands is nitrogen bases. Nitrogen bases. And there's 4. So first, we have adenosine, which we represent with an A. Next, 2, we have thymine, which is represented with a T. Our third one, is going to be cytosine. We represent that with a C. And 4, we have guanine. And we represent that with a G. All right, so now that we have our 4 bases, we're going to place them on our double-stranded helix. However, we need to know which base bonds with which base. So we know that A and T always bond together. Those bases always bond together. They never bond with C or G. They always bond together. And then C and G always bond together. So those are our pairs of our nitrogen bases. So let's draw these out. So let's draw an adenosine here, and then that connects with thymine. And then we'll draw our cytosine and our guanine. And those two always go together. All right, and these can go in any order. You can have another A and T down here, second or third. It just depends on your DNA. Everybody's DNA is different. So let's do another example of A and T. So we're gonna put T here and A here. And then we'll do another one. We'll do guanine and cytosine again. Guanine and cytosine. All right. So there's still space in between here. We have to figure out what now connects these nitrogen bases, and that's called hydrogen bonds. So I'm going to write that here. Hydrogen bonds. And later, when we talk about DNA replication, these hydrogen bonds will be important. So our hydrogen bonds are going to be in between here, connecting our nitrogen bases. Connect nitrogen bases. All right, so the rule is that Adenine and adenosine and thymine have two bonds each. So we will represent that with 2 lines connecting to them. So there are hydrogen bonds. Draw an arrow. Those are 2 hydrogen bonds for adenosine, and thymine. So another rule is that cytosine and guanine have three hydrogen bonds each. So I'll represent that with these 3 lines connecting to cytosine and guanine. All right, so let me just finish up drawing my hydrogen bonds, 2 for T and A and 3 for G and C. And that is the gist of the structure of DNA. Once all this is created, it's going to twist and look like that. That is it.